Good afternoon, everyone. We're going to go ahead and get started. It's my pleasure to introduce James Lynn. Dr. Lynn is the inaugural executive director of the Convergent Bioscience and Technology Institute at IUI or IU Indianapolis. And he's been here about 6 months. This new institute launched in August of just this last year. So the Convergent Bioscience and Technology Institute, I think we're going to hear a lot more about it today. So I don't want to steal his thunder here, but I'm really excited to hear more. And Dr. Lynn is also a professor of biomedical engineering in the Letty School of Informatics, Computing, and Engineering. And formerly, he was at the University of Cincinnati. So he's a researcher himself and also an entrepreneur. And he earned a PhD and master's degree in bioengineering and biomedical engineering from the University of Michigan. I'm going to hand it over to Dr. Lynn now to give us a little bit more information about this. And I think we have had conversations. I've had many conversations with Phaedra Corso, who's on our board, about potential collaborations with this new institute. So I'm excited for a conversation. I do have to step out 15 minutes early, and we'll have David Hagstrom help with moderating. So please be thinking of questions to ask and hopefully have a good conversation. So thanks again for being here.
Thank you, Dr. Pazzer. Do I need to stay with this microphone or use this one? Yeah. Okay. Can everyone hear me? Yeah, maybe I'll just move. Once again, thanks so much for inviting me here to join everybody, especially on this frigid weather. I hope everybody got here safely and pleasantly. So today I like to kind of divide my talk in three parts. The first part I like to utilize the opportunity to introduce you about my background, a research interest, and then moving to the last part. I think a lot of you have probably heard about Sabati, but it's still very abstract, actually, for the entire community about what we will be pursuing as our future research axis, and how we can build the collaboration with all the peer institutes within this campus. So allow me to share a disclosure. I'm a founder of a startup company called Amplicore, and there's no conflict of interest with today's presentation. So a little bit about myself. I'm originally from Taiwan. So by training, I'm an engineer, kind of a transition my training from structural engineer to biomedical engineer. As Rachel just mentioned, I went to Michigan to pursue both my master's and PhD at biomedical engineering, and then started my first academic career at the same school in neurosurgery to oversee a dedicated spine research lab. I'll touch a little bit on that later. And then in year of 2014, I managed to go across the border and to invade into the homeland of Buckeye. But fortunately, I found people there are very nice and obviously less hostile than residents in Columbus. So I spent my very first few years leading a new initiative, particularly focused on pediatric tissue engineering, and then I was brought to orthopedic surgery to oversee their new research arm 3 years later. And then also at the same time, as Rachel just mentioned, I developed a hobby to kind of create a startup. Starting from my Michigan time, we founded a company called Tissue Regeneration System that kind of translate the scaffolding technology I will show you shortly to the real clinical application. And that technology was later acquired by Bioscience, a Johnson & Johnson subsidiary. And then I kind of moved to a new territory, start to explore new drug with injectable formulation to treat vascular tissues or in the so-called sports medicine arena. So let's talk a little bit about where I started my journey by, originally trying to become a civil engineer, and then from building like a construction to a human body. So I first find a job to start my assistantship in a hospital in Taiwan. It's called McKay Memorial Hospital. And I still remember the first day I got my white coat. This is the actual image, popping on the top of my head. That's when, the TV, episode, Grey's Anatomy, hit on TV. I felt I will, look pretty sharp like this. But it turned out this is my, how my life looked like, at a hospital. I was assigned to literally wash all the prosthesis or joint implants retrieve, from the patients. So I end up washing over 200 sets of joint prosthesis. But it's not for nothing. Well, because that's the first time I got exposed to the word called biomimicking. And also, the first time to learn how you can combine the multi-discipline skill to try to address a clinical problem. So this is the system, maybe none of you are not quite familiar with, but this is the first system that Bioscience has developed to try to develop a congruent anatomy shape of the metal implant, trying to match the anatomic defects or anatomic shape of your joint. Back then, there's a big problem of wearing. The components in a joint prosthesis actually combine metal parts. That's how they actually shape your femur and tibia and mount with those metal plates. And that kind of, we call it a condyle. But and in between, they put a plastic insert to simulate your cartilage. And that's the entire motion mechanism for artificial joint. But the problem is, over time, you will have excessive motion or articulation. So you will grind that plastic parts. And eventually, you have those particles, or we call the debris, eating your bone. And you can create excessive bone absorption, and over time you have some loosening problem. That's the time when you have to do revision surgery or replace with the new pair of artificial joint. But then, originally, this design kind of creates the idea to reduce that excessive grinding or excessive wearing, because it creates those plastic parts highly matching with the anatomic profile with your joint. However, they start to realize that even though they have those plastic parts simulating your meniscus, simulating your patella joint, they still couldn't avoid that wearing problem. Then I kind of by washing those processes, I got a chance to analyze the problem. And interestingly, we found a similar parts from civil engineering we call the cantilever beam. And so I used that, the cantilever beam actually was used to convert a compression load into a bending moment. And that kind of explains why you have those split failure in a lot of kind of patella components in this, they call the low contact stress design. And that's when I start to realize, you can actually, utilize, skill sets from other disciplines and try to find out the in-depth problem, even from a clinic. So that kind of inspired me to pursue further about my kind of a future career in biomedical engineering. If we look at, the top chronic assets, that kind of show you the evolution of the so-called computational power over time. And certainly millennium or the year of 2000 is a big division. Before then, we have that personal computer and kind of, promote a lot of, accessible computational power. Some of you may still remember you have to bring a whole big stack of card to a supercomputer and then wait, for days or hours. But with, personal computer, it also drives the orthopedic surgery in the whole biomechanical analysis, including the simulation we call the finite element. And that's pretty dominant within that 2 decades for orthopedic research. But then, even although at 2000, a lot of you still remember that's when the bubble of .com just got blown off. But that created a new era of remote and parallel computing and actually facilitate molecular probing in a lot of mechanisms. Interestingly, that also when the entire orthopedic research start to move into a more molecular world, new moleculars or new growth factors were discovered, and with genetic engineering, we start to manage to create the recombinant proteins to try to facilitate that tissue augmentation. And then, then later after that, you have cloud computing, you have the AI. So we'll see, what will happen next for the entire orthopedic research. And I was very fortunate to come to the States at that critical juncture to try to catch the wave for tissue engineering. So the idea to engineer human tissue is actually not new. Ancient Egypts, they already know how to use the wood or even some glass to try to facilitate or to use that as a prosthetic support for a defective limb. But not until in 1993, Dr. Robert Langer, he's also one of the founders for Moderna, the mRNA vaccine. And also Dr. Joseph Vacandi at MIT, they start to forge all the concept for the modern tissue engineering together. So by combining, we call the tissue engineering triplets, namely cells, growth factors, and scaffold, we can now culture human tissue in vitro and then transplant back to human body to replace disease or damaged organs. However, we then start to realize there are obstacles within each element. For scaffold, it's not very easy to find biocompatible materials within to facilitate the entire scheme. And also, many of them can possess adequate mechanical function or integrity to support the initial load bearing. And also, for recombinant proteins, many times they are associated with high cost of manufacturing. And in fact, the very first few growth factors we use in clinic, including the recombinant BMP2 to facilitate spinal fusion to treat low back pain, creates a new set of complications because the recombinant protein, many times the dose is super physiological, so they can create excessive tissue formation. For cells, we all know you can have autogenous source, you can have allogenic source, but they more or less create problems in terms of how they can really receive the right cue and to really move into the right lineage. The other thing is, many times they even receive the right cue, but they don't drive into the right destination. For example, they can become very tumorigenic. But then we realized that even though it's not perfect, and there are still ongoing efforts to further polish the so-called triplet combination, and even with the new, for example, genome editing, new kind of new type of cells, including iPS cell. We then realized actually by combining the power of any of two, you can still create a kind of a new research interest. For example, we found. If you recall, several years ago, there's a kind of a big topic for cancer stem cell. We realized there's a subset cell in a tumor mass that actually possess stem cell attributes, and they share the same templates in terms of differentiation. Even although the recombinant protein can be super physiological in terms of its dose, we found like, it can actually drive the differentiation of those cancer stem cell population, and then kind of show a potential treatment as a differentiation therapy to dampen their malignancy. And we also found like, if we combine cells with scaffold alone, many times you can actually hybridize the cell with the material, and for them to become, we call the cell-responsive living composite. We call this name because like, the acronym is called SILK. So that's kind of a new type of a smart material we are pursuing because, they can replicate the cellular response and to self-regulate its material property. It could be stiffer, it can degrade faster. So it creates a new category of a research topic. And today I'm going to concentrate on how we combine growth factor with scaffold. So the scaffold alone, as I say, many times it provides immediate mechanical function, but they are inert that they can drive a lot of biological cascade. So we found like we can actually try to, by being inspired by the extracellular matrix, we realized that we can actually integrate the growth factor in those man-made material for them to become more capable in tissue engineering. So this is the very first efforts in this area driven by a lot of chemical engineer. The first thing is, again, they try to replicate the morphology of tissue. And many times the immediate thought is to create porous architecture. So they try different way. From the very beginning, they use salt. They embed salt in a polymer and then dissolve that in water to create a very random porous structure. They use the electrospinning, gas forming, extrusion deposition. But none of those are close enough to the actual tissue architecture in terms of the porosity, the distribution of the pores, or interconnectivity. So that can later create a problem in terms of vascularization, because you need those network, you need the strass, you need those porous conduit to support the vascular formation in terms of to support the new tissue. So I was very fortunate to meet my mentor, Dr. Hollister and Dr. Golston at the University of Michigan. They are considered as the pioneers for bone adaptation and remodeling. So if we look at bone tissue, it's actually a smart tissue, or as just introduced, it's actually a sealed tissue. It regulates itself. If you put them under a microscope, you will soon realize for your bone, you have a spongy bone, or we call it medullary bone. It has the microstructure of trabeculae, or you can have a very dense shell on the outer rim of a long bone. We call it cortical or cortex, the cortical bone. You then realize that cells embedded in this microstructure, they actually utilize the unique structure as their antenna to receive mechanical stimuli. And then they can make a decision where you are going to deposit the new tissue. So your bone actually remodel every day. And even after fracture, they can still reshape themselves to follow, in our term, like a principal axis of your load. So this is the most renowned Wolff's law. So we then realized if you want to create a porous structure or porous architecture that really works or that really drives functional tissue formation, you have to replicate this hierarchical relationship so that the microscopic characteristics can help possess the macroscopic tissue properties. Is that clear so far? Okay. Okay, next. I intentionally include this slide to make me look smart. So to explain this in a plain language, again, if you look at the tissue structure, many times they can be very heterogeneous and also very arbitrary. So that won't give us enough information as our target if we really want to create that structure, right? So the first algorithm we developed is called homogenization theory. So by homogenize that irregular structure, we can get an equivalent target property in terms of the permeability or elasticity as our goal. And then within the domain, this is probably to me the most relevant topic, to Regenstrief is the data. So as soon as you get the data from the acquired image, then based on the image or the voxels distribution, you can homogenize that entire tissue and to get your target property. And then the next step is how you're going to arrange your polymers, or how you're going to arrange the material you are going to use, so you can build a new structure that possess the equivalent properties of your targets. So the next algorithm we call it is called topology optimization. By giving your design domain the fixed or the targeted porosity or the elasticity, the algorithm will try to arrange your material distribution. So to give you a coarser idea about how you're going to arrange your material. But you can still get a very intricate structure of your scaffold. So we run the secondary topology optimization to get the final microstructure. Okay, a further plain language, human language, is we actually create a library of Lego, just like kids' tool or kids' toys, right? So by targeting different parts of your bone, from pelvis, from your mandibular joints, from the skull, like from your spine, we have the targeted property along all those upper bound, all those limits. So they can balance the porosity. Again, porosity supports vascularity. And also with the elasticity or stiffness, which is your rigid material part. So by this algorithm, we create those unit cells, or you call the microstructure, and then you can stack them up into a new geometry that fits in your tissue defect. So we kind of use the microstructure to build the testing sample, and in terms of regulation, FDA will be looking at how you're going to test and your bench work to make sure you meet the criteria. So we build those slots and meeting some standard we call the ASTM, and show we could by design this microstructure, we can actually meet the target properties of the human native tissue. But the next challenge is, well, if you look at the computer, they look almost like a perfect, but how are you going to replicate, or how are you going to produce those complicated structure? Back then, we didn't have that sexy name called 3D printing. But this is kind of the system that basically presents the same idea. It's called the additive manufacturing, and it's actually a bigger umbrella to cover 3D printing. So instead of doing the traditional drilling, cutting, this approach actually builds your construct layer by layer. So this system is called selective laser sintering. You can use any kind of biomaterials in powder form. In our case, we use a material called polycaprolactone. Actually, a lot of you receive dental implants made with the same material. It's called PCL. It's with a low melting temperature, so that's why, in a lot of a heating process, it's easier for you to deposit the material layer by layer. So this system actually have a powder bed, and then you can slice your final construct of your design implant. And then the laser beam will draw on the powder bed and gets that cure, and then the powder bed lower down, and then you build the second layer. And so layer by layer, you can have your construct. So I hope you can, to some extent, kind of understand how we integrate the microstructure to build the macroscopic device. So we actually use this approach to design bioabsorbable spinal cage. That's per the procedure when you have a degenerative disc disease. You have to remove your disc, but then you have to put something to maintain the disc height, which we call the spinal cage. And previously, we also used a very rigid metal, either titanium or stainless steel, but that creates excessive stress. So you can increase the bone absorption and then fail the surgery. So we actually develop a bioabsorbable cage. By just putting in that space, it will degrade over time, and then you get the bone fuse between two segments. We also develop a temporomandibular joint, and also for craniofacial repair. However, this is only the beginning. If you recall my topic, we probably successfully to replicate the morphological features of the scaffold or of a tissue. We can further promote biological response. The best we can do is to use the add-on approach. For example, we further the modification of the substrate by place a mineral layer onto the polymer and try to attract the peptides. Or we use the viral vehicle and use those scaffold as the delivering vehicle to release those viral factor. But none of those create a very sustainable or consistent the tissue formation. And so the next challenge will be how you can further those structural elements. The other thing is, as I say, today's topic, I'm going to just concentrate on how we use scaffold to address the pediatric tissue reconstruction. So this is also the first attempt. So a lot of time, you probably hear a TBM, the tracheobronchomalacia. It's a congenital defect, many times associated with newborn. So they have the defected the cartilage ring in their trachea, so they can support their trachea when they inhale. So your trachea will collapse, and then those newborn can suffocate to death. So this is something we built to address this problem. By utilizing the technology I just showed you, we actually create a tracheal splint. And then when we suture in the TBM trachea, it integrates with the original defected tissue. So far, this technique has saved over 20 babies. And the first baby received this surgery is still alive today. So as you can see, we have a number of active investigation, and I want to focus on the top two. We still have something that related to sports medicine, and also developing a patch to repair spina bifida. But today I want to continue the background story I just shared you, how we develop the bone structure, and how we can further our efforts in reconstructing human trachea. So some take-home message from the first part of story. We managed to create a unit cell as the very fundamental element for the modern scaffold to support tissue engineering by utilizing the algorithm we developed. And then, we introduced the additive manufacturing that we can really replicate and make this intricate scaffold or construct possible. However, they are biomechanical sound, meaning we only retain a portion of its function in biomechanics. But we really need to further its physiological function and also trigger those biological cascade. So if you remember my first story, about that joint story, we observe, what actually happened in a human body or in nature. And that's the same thing. When we look at our approach to make a scaffold, we rely on those struts or bundle, if you will. However, they don't have any biological cues to drive the cell component for them to become a viable tissue. So something is missing here. We then realized if you look at the ECM, they provide a structural bundles or structural fiber with those biological or proteins embedded. Is it possible for us to also follow the same paradigm and design a more functional scaffold? So with that kind of a attempt or question in mind, we then try to see if is it possible to create a composite scaffold so we can use the best of man-made material or synthetics with biofactors as a signal to make sure, we can make the meaningful, the phenotypical expression of the target tissue. So here's our approach. As I just showed you, many 3D printing or additive manufacturing, they are driven by heated process, which creates a very harsh environment for your biological component. To protect this biological component, we have to create a primary carrier for them first. So we decellulose matrix, just like the same protocol that has been prevalently used by a lot of tissue banks in this country by kind of washing out the pathogen or remove the cells, we can get the decellulized tissue as our graft. And then we develop a technology to encapsulate those tissue. And the approach we develop is we melt the polymer mallet and also mix with this microspheres or micro beads, and then extrude them into a filament. So you will have those beads that contains human or animal tissue in those printable bundles or printable filament. So the first thing is, we have to make sure we can retain all the biofactors we need before incorporating them into the structure filament. So after that process, we show, even although we probably change the distribution or the spectrum of the proteins or biofactors we need, but we still preserve most of them after the tissue processing. And the second part is we then try to show, even we encapsulate those tissue, those growth factors or those cytokine can be still released from the microsphere. So the right panel showed the microsphere contained with the processed tissue. Even you see cells in the basal media, you can still make it to present the same results as you see in the post media, which is chondrogenic. Then we develop a differentiation assay by seeding the human mesenchymal stem cell palette. And then in the media where you also have those microspheres releasing the growth factor and see if they can drive the chondrogenic differentiation, which is the process for cartilage formation and development. So the results show that, again, remember when I talk about the tissue engineering triplets, many times the biofactors can be super physiological or overpausing your cells. However, that's the protocol, I learned when I was a student. It's almost like a standard. But then we realized if you pause a stem cell for them to become chondrogenic, they won't stop. It's kind of the same situation as the long bone formation. We know like the bone formation has that kind of two types. One is called endochondral ossification, meaning like you will form cartilage, and then the cartilage will further calcify, or you have a hypertrophic chondrocyte, and then they become bone. This shows that if you use the paucity media or chondrogenic, that will be the case. You can pause the chondrocyte formation, but then they can stop there. They can become more hydrophobic and then become a bone cell. However, if you see them in the microsphere that contains with the process cartilage, they will just release the adequate or sufficient chondrogenic factor for them to become chondrocyte and then stay as is. So we then try to incorporate this microsphere into the bundle. And again, this is inspired by how the ECM structure looked like. And we used alkaline phosphatase, which is the bone marker, as a surrogate, and to show even although this tissue is embedded in the bead and then kind of a mix with the polymer to become the bundle, they can still release enough ALP just as if they release from the microspheres alone. So this is a busy slide, but I'm just trying to show you the result we observed. So we know like a chondrocyte, our cartilage, they like to be formed in a low oxygen level, or we call hypoxia. And the key is for those chondrocytes to form micromasses or a spherical aggregates, so they can maintain the low oxygen environment, and that is more favorable for cartilage formation. So with the scaffold embedded with the microsphere, with the ECM, you can see those micromasses formation onto the scaffold compared to scaffold without those microsphere. And then again, we show if you use the chondrogenic media to pulse these cells directly, they won't stop at the chondrogenic phenotype. They will continue, they will march to bone formation or osteogenesis. The next is we want to see how we can translate this technology by building, use this Bioinspired filament to create a cartilage scaffold to treat the actual, we call it osteochondral lesion. You can imagine it's just like the dent on your car. A lot of active young athletes or active kids, they can ride a bike or they can do a lot of sports and then hit their knee, that creates a dent directly on their cartilage. Unfortunately, the cartilage barely heal themselves because of the low vascular, or we call it avascular tissue. So the current clinical treatment, including a lot of a transplant technique. One of that is we call it OATS, or osteochondral autologous transplant surgery. The surgeon will prepare plaque from a non-articular surface, which is many times a little bit superior from the articular surface. And then they prepare those plaque, and then they, at the defect site, they kind of, poke few holes and then transplant those plaque there and hope, they get union by themselves. Unfortunately, this surgery still with a significant failure rate, because the cartilage itself won't get union, so over time the graft will get peeled off. We need a better approach to make sure we can form a very intact cartilage. So we then introduce our scaffolding technique and also see how the embedded microsphere with the decellulized tissue will do their job. Again, the first thing as an engineer, we always care about how capable your scaffold can provide the load bearing, because we know, those are large joints. They have to provide initial support. So we show that with or without those microspheres, we won't compromise their mechanical strength. And with those embedded beads, we can actually accelerate the degradation of those polymers with a more controllable release compared to the microsphere alone. And again, we show, again, I'm just going to show the key message here. With the microsphere that contains the processed tissue in the scaffold, you can actually facilitate the formation of micromasses. That's a kind of indicative sign for the local cartilage formation. Are you still with me? Hopefully, won't get lost. So that's kind of the sign, many times we are looking for in our in vitro cultural system, is to show you have that micromasses formation. Again, compared to the current standard to culture cartilage in porcine media or chondrogenic media, they can drive the hypertrophic formation, or driving those progenitor cells to become bone cell or osteoblast. Whereas if you just use our a substrate, meaning scaffold with microsphere that contains the processed cartilage, you can provide just adequate signal and to make them maintain the chondrogenic phenotype. So this shows you why this happened. So if you provide just adequate cues to your progenitor cell, you will trigger a translational pathway called SOX9. The SOX9 will drive this progenitor cell to stay as chondrocyte, the kind of the progenitor cells, for cartilage formation. However, if you over-pause them, in this case, you use the pausing media, or as we say, you kind of give a too excessive a supplement, and they can continue to drive into a transcriptional pathway called RUNX2. That's the differentiation pathway for bone formation. So the conclusion is that instead of following the traditional triplets for tissue engineering, we actually develop a better approach to maintain the persistent chondrocyte formation or the chondrogenic expression. So by including those the processed tissue into your scaffold, we can replicate the features from the extracellular matrix and drive the right tissue formation. So this is how we show in our large animal model. And again, I just want to show you that compared to the oats, where the current clinical standard is, we actually show with this approach, we can have a better interface formation. You need to form a polarized cartilage to retain the water. And this is a key to show you that the functional osteochondral interface is there. And this is what we are pursuing, because many times the joint replacements, it's not the right indication for a lot of young kids because they are still growing. So over time, we want to translate this technique to become a joint preservation technique to substitute the joint replacement for younger patients. To wrap up, so for osteochondral regeneration and integration, the first generation of scaffold can only provide structure. We then further incorporate the microsphere technology with the decellularized matrix, so we can utilize those decellularized matrix to just provide the adequate cues to drive and maintain the chondrogenic expression. This addresses the limitation of supplies and also prevent the second donor site mobility, as we show you in the oats procedure. In the last few minutes, I want to show you some our early work in how we are going to further the trachea reconstruction. As we just show you, the story published in the New England Journal show that we can develop that splint, the tracheal splint, to support the cartilage ring defect in the tracheobronchomalacia cases. However, it won't help reconstruct the whole trachea, particularly in the long segment trachea. However, in the pediatric complication, many times you can have a pathology that include congenital defect, trauma, or malignancy that require long segment tracheal reconstruction, which is still an unmet need in the real world. So we thought about currently, like you can have a lot of xenograft, meaning you use trachea from pigs, and you process them and store in the hospital. And at the time you need the tracheal implant, you use them. The downside is, they are xenograft, so there's a compatibility issue. The two is, you don't have a dimensional compatibility. So we kind of a hybrid the technology we show you with the Bioinspired filament that can print the construct, and we try to combine that with the current xeno transplant. So I'm going to skip, some details, but it's more challenging because this time we need to build a more complex layer. If you look at the trachea, the most challenging functional layer is mucosa. So this time we need to integrate and also build a more complex layer and also seeded with different microsphere that contains different tissue. We develop a spiral slicer to address the dimensional limitation. So we can slice the xenograft, and also when we build the counterpart of the spiral, we can now strain them together. So we can build kind of your implant with any kind of length, or with the any type of diameter. You kind of you can kind of roll them together, and so you can build it longer, or build it wider. So this is just start. We under the SEM, we show our microsphere with a different part of a tissue can be embedded in a different layer, simulating the overall construct of a trachea. And these implants have been put in our pig model. So the next step is we will see how far they can go in terms of to build those complex layers. Okay, in the next 3 slides, I would like to show you why, this great city is trying to bring in a new innovation hub. I was very fortunate to experience several different transformation of the innovation district in Ann Arbor. Some of you may know Pfizer used to have one of their global R&D center in Ann Arbor, but then they decided to leave. So University of Michigan kind of flipped that entire campus into a new innovation hub. However, because there's no industry support, even though it's in a pure academic setup, it won't grow very far because there's no strong industry support. In Cincinnati, it's the opposite case. We have some strong industry partner like Procter & Gamble, Ethicon, but we don't have enough land to grow that. In Cincinnati, we create something we call Cincinnati Inno District. We actually flip a Sears department store. So it only provides some infrastructure for kind of for collecting ideas, but it's very difficult to really translate those ideas and then really lend the startups or to promote commercialization. Columbus, they are doing a great job to lead the 6-wave gene therapy because they have a very strong urban university in their capital, and although they don't have a very strong industry partner, but they have their strong clinical partner, which is the nationwide children's hospital system. We in Indianapolis, which is one of the reasons attract me to move my career here, is we pretty much collect the entire momentum by gathering everything like I have just mentioned. We have strong industry partner. We have a strong urban research university, which is this new IU Indianapolis. We have probably the largest medical school system to support this. So there's no reason this place won't just get thriving. So to build this conglomerate innovation hub, we need a powerhouse, especially after our recent separation from our peer, Purdue University. We are really eager to develop our in-house capability to support a lot of health innovation. Therefore, for the current CBATI, we set 4 pillars. The first pillar is the sensing technology. The second one is new implantable. That's kind of aligned what I have talked about so far. The third one is molecular measurement. And the fourth one is smart drug delivery, kind of aligned interests with our strong partner, Eli Lilly. So as you can see here, we have created a pipeline joint search for our faculty members, and we try to land about 10 to 12 faculty members for CBATI in the next 5 years. The 4 pillars actually form 4 quadrants, and that kind of shows you the intersections of their expertise. We have a current search for wearable and implantable detection, and also one for computational biology. And aligned with the interest from School of Medicine, we are currently also hiring a regenerative biology and medicine, as well as an endowed chair in physics, specialized in optics and quantum characterization. The ultimate goal is we are trying to work with all the IU transdisciplinary or multidisciplinary research institute to build a big cluster. I call it EPIC, to try to propel the health innovation. My institute will be, again, specialized in promoting those 4-pillar technology. And we try to land a center grant from NSF. It's called Science and Technology Center, SciTech Center. The idea is to try to grow or develop the next generation of materials to support those 4 pillars. Or in other words, we want to develop a new class of silk. If you still remember what silk means, it's called self-regulating living composite. It's smarter. It can sense all the stimulation from the environment and then improve its property. We also have another institute within IU Indianapolis called Institute for Human Health and Wellbeing, or H2W, and working with other existing successful units, including the ICMH and also IBRI, which is leading another initiative called the Center of Excellence for Precision Medicine. We believe this will be happening and also promotes the overall health innovation in this great city. I guess I'm going to stop here. Sorry, I'm running out of time, but please just share your thoughts and questions. Thank you.
Sounds like you're doing great work with a lot of real-world possibilities. Have you taken this technology into an in vivo subject at this point? And so the 3D printed framework plus the decellularized plus, okay. What have you found?
So there are 2 parts. For the structural components, we have actually conducted clinical trials with those because FDA also clears some 3D printed implants for non-low bearing. So as I say, it provides a new category of supporting construct to substitute metal implants.
Yeah.
And as I say, like, you create a new set of complication with the metal implants. And our 3D printed scaffolds certainly address with that, but it's more passive. So at this point, like, it has been validated with those structure component or structure feature. But at this point, like, we are still at our journey to incorporate those biological tissues. For example, like, we have used a pig model to show the osteochondral repair, as I just showed you, like, at the very last slides. But it's not moving the human yet. Okay. But for the pure 3D printing scaffolds, it's already, like, in clinic.
Yeah. And one quick detail question. The optimal shapes that optimize permissivity and bulk modulus, do you use only one shape when you're building the matrix, or do you use combinations of shapes to those Lego pieces that you talked about? It wasn't clear whether it was just a homogeneous or whether it's heterogeneous with multiple shapes.
Yeah, so the idea to build the library or the Lego units, it's, it won't be constrained with more global geometry. Meaning, like, for example, if you have a long bone defect, or if you have a hole in your pelvis, you take that as your design domain, and then you use your Lego, to put in that larger geometry. Then that would be your final implant.
But do you use multiple Legos or only one kind of Lego when you create?
You can use multiple. That's a great question. Depending on the complexity of your tissue. For example, like, for trachea, you can actually use 3 Legos to construct or constitute your complex tissue structure. For bone, in the real world, we actually build something we call biphasic. If you look at the joint, I show you, like, the plaque, you have a cartilage on the top, and you have the bone underneath. We call it subchondral bone. You can actually pick 2 Lego. One, it's your kind of a cartilage Lego with your, like, a bone Lego underneath. So you build a biphasic scaffold as your new plaque, and then put in a hole.
Are you familiar with Thingiverse? Online? No, it's an open source repository for 3D printed shapes. I was going to make the funny joke of saying, have you posted these to Thingiverse yet? Which is where a lot of home 3D printers go to get their shapes.
Yeah,
so
maybe that's a new business, right? I provide the Legos, for all kinds of hobbies. Yeah.
Well, I'm going to speak into the microphone here too, so that you can hear me. We're about at our time. I just want to make a comment and an invitation. So you mentioned at some point in your talk the Regenstrief's reputation for data. And I mean, a lot of the types of data we commonly work with are clinical data that comes through the routine course of clinical care delivery, medical data, some, relevant data sources that you perhaps touched upon were potentially imaging data, pathology data. So, as an organization, we're interested in engaging scientifically with partners, as well as potentially in this entrepreneurial space that you're describing. So consider that in the future. And thank you so much for sharing the, really amazing and impressive work you've already done. And thanks for visiting us.
Oh, thanks so much for the opportunity for me to share this. Thank you very much.